Hello viewers, welcome to our channel Biological Concepts. In today's video, we will discuss on nonsense mutation, a type of point mutation. Definition, examples, diseases caused by nonsense mutation will be discussed here. So stay tuned. Now first of all, we'll discuss the definition of nonsense mutation. A nonsense mutation is a point mutation in a DNA sequence that results in a nonsense codon or premature stop codon in the transcribed mRNA and leads to truncated, incomplete and possibly non-functional protein product. So basically nonsense mutation is a type of point mutation. It's a type of point mutation. Now what is point mutation? A point mutation is the genetic mutation where a single nucleotide base is changed or inserted or deleted from a DNA or RNA sequence of an organism's genome. So, the point mutation may be either substitution type of mutation or insertion deletion type of mutation. Where it is substitution type of mutation where a single base is changed. Base is base change. Base change. Single nucleotide base change. Inserted or deleted. So it may be either substitution type of mutation where the base is changed or insertion type of mutation or deletion type of mutation. Here this nonsense mutation is the substitution type of mutation where the base is changed. And this change results in nonsense codon or premature stop codon in the transcribed mRNA. Now what is codon? A codon is a sequence of 3 DNA or RNA nucleotides, that is, it is a trinucleotide. Codon is a trinucleotide. Codon is a trinucleotide. And this trinucleotide may be either DNA nucleotide or it may be RNA trinucleotide. In case of DNA, the nucleotides are A, T, G, C. Any of the 3 combination of this nucleotide will be the codon. And in case of it is A, U, G, C. Here any of these 3 nucleotides combination will be the codon. Look at this, both in case of both DNA and RNA, almost the nucleotides is same, but in case of DNA it is T, whereas in case of RNA this T is replaced with U, and A stands for adenine, T is for thymine, G for guanine, C for cytosine, and this U for uracil. In case of RNA, thymine is not present, rather uracil is present, whereas in case of DNA, thymine is present. When it is transcribed into mRNA, this thymine will be replaced with uracil. Okay. And we know that DNA produce RNA through transcription, and from RNA protein is produced through by the process is translation. From DNA to RNA it is transcription, and from RNA protein it is translation. Okay, so when DNA is produced RNA, it is the process is transcription, and RNA produce protein, it is translation. So this we have discussed all the codon. So this formation of nonsense codon and premature stop codon. So this trinucleotide codon there are 64 trinucleotide codon. Among these 61 specify amino acid and rest 3 does not specify any amino acids. This are stop codon that stop or halted the protein signal, protein synthesis. So there are 64 codons, trinucleotide codons. Among these, 61 is produce specific amino acid and 3 generate stop signal. Okay. And due to this formation of this nonsense codon or premature stop codon in the transcript RNA, it leads to truncated, incomplete or possibly non-functional protein product. Okay. Now what is nonsense codon? We have already discussed on codon. Now what is nonsense codon or stop codon? There are 3 kinds of nonsense codon. One is UAA, second one is UAG, and third one is UGA. UAA is known as ochre, OCHRE. UAG is amber, and UGA is opal. Okay, these are 3 stop codon or nonsense codon. So, stop codon or nonsense codon is a sequence of 3 nucleotides. These are 3 nucleotides: UAA, another one is UAG, and third one is UGA. This is also trinucleotide in mRNA. Why it is mRNA? Because U is present only in mRNA, not in DNA. Okay, uracil is present in case of mRNA or not in DNA. In DNA, it will be the U is thymine. The thymine is replaced with uracil in case of mRNA. So, this nonsense codon is a sequence of 3 nucleotides in mRNA that signals a halt or stop signal to protein synthesis in cell. This, when there is a formation of UAA or UAG or UGA in the mRNA sequence due to mutation, it signals to halt a protein synthesis in the cell. Okay. Normally, when protein synthesis stop, where these 3 codons are present, either UAA or UAG or UGA. Only these 3 types of codon is not specifying any amino acids. It signal the stop signal. Okay, either of these 3. Now, due to this formation of this nonsense codon, the protein synthesis stop. There is premature chain termination, or there may be non-functional protein product. If there is formation of non-functional protein product, it will be harmful to the cell. So, but not always this nonsense mutation are harmful. The functional effect of nonsense mutation depends on many aspects, such as the location of the stop codon within the coding DNA. For example, the effect of nonsense mutation depends on the proximity of the nonsense mutation to the original stop codon and the degree to which functional subdomains of the protein are affected. Suppose this is a 5' to 3' mRNA, and there will be the stop signal nonsense codon that stop the protein synthesis. Okay, here protein synthesis halter. So here it is mentioned that the effect of nonsense mutation depends on the proximity of the nonsense mutation to the original stop codon. So this is the original stop codon. So if this stop codon occur at that position just before the stop signal, in that case it is not so much harmful. But if this stop signal is here or here, so in that case the protein synthesis will be stopped here or here. So it will be very first the protein synthesis will stop. Okay, in that case it will be very harmful. And another case, if suppose this is a functional subdomain, or this is a functional subdomain, and if there is on this here there is a formation of nonsense codon, or this is also subdomain. Among here in this subdomain, if there is formation of nonsense codon, there also protein synthesis will be stopped. So if the nonsense codon form in the functional subdomains, so there the functional subdomains, in this functional subdomains the protein synthesis will be affected, the protein synthesis will be halt. So in that case it will be may also be harmful. Okay, but if the stop codon is from somewhere in the location where the functional subdomain is not present, so in that case, so the nonsense mutation is harmful. Okay, next is, as nonsense mutation leads to premature termination of polypeptide chain, they are also known as chain termination mutation. So this nonsense mutation also known as chain termination mutation, as it leads to premature termination of the polypeptide. It leads to premature termination of the polypeptide. So this nonsense mutation also known as chain termination mutation. Okay. Now let's discuss about the examples of nonsense mutation. So this is the original version. This is the original. We just write like this: original. Means original version, and this is the mutated version. Okay. In the original version, the DNA sequence 5' to 3' DNA sequence is 5' ATG, ACT, CG8, AGC, TG8, 3'. And as you know that A paired with, this is double-stranded DNA, and A paired with T by 2 hydrogen bond, and G paired with C by 3 hydrogen bond. So the 3' to 5' DNA sequence is A paired with T, T paired with A, G paired with C, then TGA, GCT, TCG, and ACT 5'. So, and the DNA through transcription produce the mRNA, and the 5' to 3' mRNA sequence is like AUG, ACU, CGA, AGC, UGA. Look at this. Both the sequence of the 5' to 3' DNA sequence and the 5' to 3' mRNA sequence are same, except the T is replaced with U at the same position. As you know that in case of RNA, the T is replaced with U, that is thymine is replaced with uracil. So both the 5' 3' DNA sequence and 5' to 3' mRNA sequence are same, except the thymine is replaced with uracil. Okay, and as you know that mRNA is synthesized in 5' to 3' direction. Note that. Okay, so this, we just consider this trinucleotide. Okay, this CGA produce the mRNA where the CGA is the produce arginine. It's specified arginine. CGA. It specify arginine. Okay. Now at that position, this C is replaced with T. Okay. So the GCT also replaced with ACT, and the mRNA, in case of mRNA, this CGA is replaced with UGA, and you know that there are 3 stop signals that are UAA, next one UAG, third one is UGA. Any of these 3 will generate stop signal during protein synthesis. As here UGA is produced, so it also generates the stop signal here. So it is also nonsense mutation. Okay. Now the DNA sequence CGA, in the DNA sequence CGA causing cytosine, causing this cytosine to be replaced with thymine. This mentioned that it is the original version and this one is the mutated version. So this C is replaced with T, that is cytosine is replaced with thymine, leading to TGA in the DNA sequence. So CGA is produced TGA, CD is converted to TGA. This results at the protein level in the replacement of arginine with stop codon UGA, because in mRNA sequence it is CGA in the original version, whereas in the mutated version it is CGA is replaced by UGA. So this UGA is the stop codon. So this arginine is replaced with the stop codon UGA. So this one is also, this is the nonsense mutation. Now let's discuss the diseases which are caused by nonsense mutation. There are several rare genetic diseases which are caused by nonsense mutation, and these are hemophilia, Duchenne muscular dystrophy, and cystic fibrosis, and also frequent diseases such as cancers, neurological disorders, and metabolic disorders. So these are all about nonsense mutation. If you like this video, please like, share, and subscribe to this channel. Thank you.